think we'll do is we'll go straight to Dr. Alberto Catalano, who's going to speak to us about the laboratory hematology, the science behind the scene. So Dr. Catalano is the principal scientist of the Institute of Haematology, Royal Prince Alfred Hospital. And he has, in fact, been a research scientist for many, many years and continues to do that research scientist while also being the principal scientist of our service laboratory. So he's someone that we cannot do without in our unit. But as a result of these years of experience, and now not only as a research scientist, but also as laboratory manager, he's well qualified to look at the laboratory investigation of hematological malignancies, with his major interest being in the molecular biology of myeloid malignancies. Thank you very much, Albert.
Good afternoon, everyone, and thank you for persisting with me. I'd like to welcome you to my talk, which I hope will not blind you too much with science, and may explain some of the behind the scenes things about the lab. So firstly, I'd like to say that Dr. Christian Bryant really well covered flow cytometry. So I will talk about flow cytometry, but from a different aspect, I suppose, explaining how it's actually relevant from the biological side, and also talk about basic things like how your full blood count is determined, and how we do morphology, and then talk about molecular hematology and a little bit about coagulation. So all the main things in the lab that we do and cover that are relevant to blood cancers. So blood, starting off, is a mixture of cells. And it's a mixture of cells and liquid called plasma. And this is all circulating. And when you go to get a blood draw, we get that mixture taken out of a vein. And it contains red cells, which these disc-looking things, which are really quite tiny, platelets, which are even smaller than the red cells, and a whole lot of different types of white cells, as well as the plasma, which is the liquid in which they float around in. And you get the blood drawn in these tubes, and these tubes are special tubes. They've got a vacuum in them, which allows the blood to get sucked into the tubes. And for hematology tests, most of the tests that we do in hematology require the blood to remain liquid. And so the tubes contain anticoagulants, chemicals that stop the blood from clotting. So the hematology tubes are these green, blue, and purple tubes, and the biochemistry tubes are these yellow top tubes. And the biochemists like to have their blood clotted because they're looking at things in the plasma, or in the clotted blood, it's called serum. And they're looking for, proteins and other components that are in the serum. But in hematology, we're looking at the liquid component, and so we want the blood to remain liquid. And so one of the tests that we do is an automated full blood count. And this instrument here, in fact, there's 3 instruments there. There are 3 full blood count analyzers and a slide maker stainer that are hooked up in a train like that. It is exactly the sort of system that we have in the hematology lab at RPA. And the automated counting machines can count millions of cells in a drop of blood and determine the number of red cells, platelets, and white cells in a fixed volume, in addition to measuring the hemoglobin that is present in a fixed volume of blood. And if the software in the blood analyzer identifies something unusual in the count, it can't differentiate the main subtypes of white cells, a blood film is made. So this last part of the train is a slide maker stainer, which takes a tiny drop of blood and smears it on a glass slide, and then stains it with the stains we need to be able to see the different types of cells with a microscope. So how does the analyzer analyze those cells? So similar to flow cytometry, as Dr. Bryant was talking about, the cells flow through a channel, and then there's a detector. And this detector in the type of analyzers that we have mainly uses a combination of a laser to shine a light through the cell, and also different types of electrical signals. So it looks at impedance, which measures the volume of the cell, and conductivity. So it actually beams through a radio frequency signal through the cell, and then measures the radio frequency that comes out the other end. And that measures conductivity within the cell. And in addition to the light scatter with a laser, we get a combination of all three in that detector. And what we get is what's called a VCS plot. And so the different measures on those three different channels on the analyzer are plotted in a 3D plot. And that allows the cells to be grouped together according to the measures that were measured on those three parameters. And the three main types of, the five main types of blood cells can be classified by how they appear in that VCS plot. And so here we can see four of those five main types of blood cells. We can see the lymphocytes in blue, the monocytes in green, the neutrophils in purple, and the eosinophils in this orangey color. So each little dot is actually one of the events. So one of the cells that has gone through one of those channels, and based on the measures of those three parameters, it gets plotted on this plot. And then it allows us to count how many eosinophils, how many neutrophils, how many monocytes, and how many lymphocytes are present in that, that particular volume of blood. Now, as I said, if the analyzer finds something unusual, it makes a blood film. So the blood film is basically just a smear of blood on a glass slide. A very thin layer of blood is spread on the glass slide. And unstained blood, when you look at it under a microscope, looks like this. You can see the red cells as these little sort of pinkish orange ghosts. And then the white cells, well, under the microscope, unstained, actually look white. And there's a platelet there. So that's really hard to interpret. So we employ stains, which are called Romanowsky stains, which are basically a mixture of a blue stain and a red stain, so that we get better staining of the red cells, as you can see here, but also we get purplish colors, and we get the platelets staining up as well. And it makes it a lot easier to see the cells under the microscope and just distinguish what they actually look like, and how the nucleus varies in the cell, and so on. And so you can see from this actual photograph that you can see platelets, you can see a neutrophil, you can see a lymphocyte, a monocyte, a basophil, a neutrophil, and an eosinophil. And as I said, there's two different types of stains. So what we call basophilic stains, which are the blue stains. And so a basophil has a lot of blue staining, and that's why it's called a basophil. An eosinophil has a lot of eosinophilic stain, which is a red stain, and that's why it's called an eosinophil. And the others basically have a mixture of the two. And so the nuclei stain purple because they both stain with both stains. And then depending on the type of cell it is, so lymphocytes tend to have more bluish cytoplasm, and the neutrophils tend to have more pinkish cytoplasm, and so on. And so they have different shaped nuclei, which is where the DNA is stored in the cell. And based on those structures, the morphologist and that looks at the cells under the microscope can count the different types of cells that are present. And so we classify them under five main types: lymphocytes, neutrophils, monocytes, eosinophils, and basophils. And you can see the red cells and platelets can easily be seen. But those white cells are the important things in blood cancers. And so The mature white cells are the ones that are down the bottom, and in fact, the mature platelets and the mature red cells or erythrocytes are the ones that are down the bottom. But the immature cells are the ones that you mostly see in the bone marrow. So you don't usually see these in the blood. So if immature cells are seen in the blood, that's an indication that we need to do some further investigation. So if the morphologist sees some of these more immature cells in the blood, that sort of triggers that we need to do something. So they look very similar under the microscope, but they are very different because they are starting to differentiate and give rise to different types of cells. And so the way that we can differentiate them is with flow cytometry, because they're starting to show different proteins on the outside of the cells. And that's probably the best way to differentiate what they are. So if we do see some of these on the blood film, the next step is probably to look at the blood with flow cytometry. So I'm just going to cover some basic biology. So here's a sort of a drawing of a typical cell. And as I said, a cell has a cytoplasm and it has a nucleus. And the nucleus is where the DNA of the cell is stored. But in addition, there's the mitochondria, which is sort of the powerhouses of the cell. And so different types of cells have different amounts of mitochondria, depending on what sort of function they have. And they have messenger RNA. So the DNA genes are copied into RNA copies, depending on the type of cell. And so the different types of RNA that you see in a cell is also differentiates the type of cell that it is. And those RNA molecules then encode proteins that are either expressed on the cell surface, or they're expressed in the nucleus, or they float around in the cytoplasm. And so we have intracellular proteins, which can be in the nucleus, or cellular protein, or surface proteins that we can identify with flow cytometry. So if we add coloured antibodies, as Dr. Bryant has described, we can see what proteins are expressed on the cell surface. So here we've got a green antibody and a red antibody that are specific for different types of proteins. And you can see that this protein has a different structure to this one. And those antibodies are able to bind specifically to the protein based on its structure. And this is the flow cytometer that is used to measure that. So how that works, I think Dr. Bryant has covered, but I'll just cover that again. So as you can see, there's a little animation here. So we have a stream of red cells going through in the cytometer. And there's a laser, a very powerful laser that shines through that stream that is created by the fluidic system. And there are individual droplets, and each droplet will have a single cell in there. And if it fluoresces, you can see, depending on the color of antibodies that are present on the outside of those cells, it'll fluoresce different colors. And they're detected by the different detectors here and different filters which split up the fluorescent signal based on the colors that are fluoresced. But in addition, we have forward scatter of the laser based on the structures that are present in the cell. So the complexity of the nucleus and the structures in there, like the mitochondria and so on, which can sort of spread the light, which will give you some forward scatter and also side scatter, which is also detected on the side of the detector. And it's that combination of data which then is able to be analysed in the flow cytometer. And so here we see an image of, we've got side scatter and forward scatter, and millions of counts there. Each little dot is basically one of those droplets with a cell in it that's been analysed. And here we've got basically just two of those antibodies. So CD3, which is specific for T cells, would have a particular fluorescent label attached to it, and CD19, which is specific for B cells. Now, the lymphocytes under the microscope look very similar, and it's very hard. Well, it's probably impossible to differentiate what a B cell and a T cell looks like. They look very similar. But with flow cytometry, we're able to count the T cells and the B cells as separate populations. And you can see they're very clearly separated. And you can gate then either by drawing a circle around the cells or even just adding some crosshairs there and just separating out. And then the computer can then tell you what proportion of all the cells in this big box are expressing CD3 and are in this group, or what proportion are expressing CD19, which indicates that they're B cells. And you can get a count and percentages and so on. And that's what we report. So there's other information in the cell. And as I said, there's the nucleus, which contains DNA. And in diseases like leukemias and lymphomas and myeloma even, we get mutations, and those mutations are encoded in the DNA. And so what we get is information that is normally in the cell becomes misinformation, and we call those mutations. And as I said, the DNA is copied to RNA, and the RNA is read by cellular proteins called ribosomes to make other proteins that are expressed by a cell. And those proteins can be, as I said, intracellular or on the cell surface, or even proteins that are exported out of the cell. And so those mutations then give rise to proteins that have altered function. And it's that altered function protein which actually is probably giving rise to the disease. And so we want to look at DNA. And as Professor Ireland showed, DNA is present in the form of chromosomes. Now, each cell contains over 6 billion base pairs of DNA per cell. So here's a diagram of the DNA helix. And you can see in the helix, there's these bits that form these bridges between the two main strands, which we call the DNA ladder. And they're the bases which form pairs. And it's those pairs of bases that are critical to giving the code that defines the RNA and then the protein that is expressed. Now, here's a trivial thing. So every cell in a nucleus will have 2 meters of DNA packed in tightly in the cell. When the cell divides, that DNA contracts and forms these structures, which we can see here, which are the contracted chromosomes. And they can, as Professor Ireland showed, you can take pictures of those chromosomes and then sort them according to size. And here we have chromosomes that have been labeled with fluorescent probes, and the different chromosomes labeled with different mixtures of probes so that they appear different colors. And you can use that in cytogenetics to actually look for things like translocation. So you can see here that we've got a translocation on this chromosome there. So the DNA contains natural variation. So each individual will have a variation, and that's what makes us all unique. So we have natural variations which occur approximately every 1,000 base pairs in the DNA. But it's the unnatural variations that we're actually interested in. And so to read the sequences from the blood cells, we do something called DNA sequencing. So DNA is extracted from the nuclei of white blood cells in a blood or bone marrow specimen, and specific parts of the genome are isolated by PCR amplification. So PCR is something that we're probably all familiar with over the last few years. We've probably all had PCR tests for COVID over the last few years. But we can also use PCR to look for the DNA of our own genome, and in addition to looking for viruses that have invaded our system. So the key to all of this is the base pairing, as I said. And so the DNA strand is made of 4 different types of bases: A's, T's, G's, and C's. And the A's pair to T's, and the G's pairs to C's. And it's that pairing which is crucial to how we read the DNA. So here we have a cartoon. So genomic DNA. So here's a gene, a typical gene. And so the gene is formed out of different parts. So the thick parts here are basically the parts that will encode a protein. And these bits are the bits that are, in addition to the parts that encode the protein, also part of the RNA. So we call that the transcribed sequence. So it becomes spliced together, where these little thin bits are cut out by splicing machinery, which splices the RNA to form mature RNA, which is shown here. And that's a combination of what we call exons, which are these bits here, spliced together to form the mature RNA. And in the mature RNA, this thick part, it's not actually thick, but I mean, as it's drawn in this cartoon, is the part that encodes the protein, and the rest of it is part that is actually needed by the ribosomes to actually read the RNA and to form the proteins. So we can design primers for a particular target in that gene. And so the primers are short, synthetically generated bits of DNA, which can hybridize to specific regions of the DNA, or copy DNA in this case, that flanks the target sequence. So we heat the DNA, and that separates the two strands. So here we've got all the bases shown like that, separated by heating. And we apply the primers, and based on the sequence that they can pair up with, we can actually then amplify. So we add a protein, which is called a polymerase, which can then add the individual bases to those separated strands of DNA and make a copy of the DNA. So here we've made one copy of each of the two strands. And if we repeat that process, add the primers again, the primers hybridize, and then the protein extends that, and we make 4 copies. And we repeat that process again, and we make 8 copies, and so on. And so that with 10 cycles of PCR, we make about 1,000 copies. With 20 cycles, we make about 1,000,000 copies. And with 30 cycles, we make about 1,000,000,000 copies. So we make an invisible amount of DNA become visible, and then we can do something with it, such as sequencing. So here we have some amplified DNA, and then we've added a primer, and then we add some components to it to sequence it. And what the difference is that we have fluorescent bases that are added, which actually stop the extension of the strand, and then we separate those fragments as they're created according to size, and then we can read the sequence. And so here we have the primer here, and then we have some unknown sequence, and then based on what has been added, and as you read it according to the way that it's been separated, we can see that the first base that's complementary to the A is a T, and then the next one is a C, which is complementary to the G, and the next one is another C, complementary to the G, and so on and so on. And so that we can read the sequence and then determine what the sequence was of what we call the complementary strand. So next generation sequencing is different to that, in that instead of just amplifying one target, we're amplifying thousands of targets simultaneously. So we have thousands of PCR primers, and then we make what we call a library, and then we add them to the sequencer. And Dr. Bryant showed you a picture of the sequencer with its pajamas taken off, and you could see all the insides of it, but that's what it looks like from the outside. And so we put the library in what's called a flow cell. So it's basically a sandwich of 2 pieces of glass with this channel there. And in that channel, we have what's called basically oligonucleotides, which are other types of primers that have been attached to the glass. And then the library, the amplified pieces of DNA are able to attach to the glass slide. And then there's a camera in the sequencer, which is able to read the fluorescent signal as each piece of library basically is amplified. And so we have basically a camera that sequentially reads as each base is added one by one. And then we can read if there's a T present in a particular spot, or a G, or a C, or another T, or another A, or another C, and so on. And then we can sequence millions and millions of pieces of DNA all at once. And they generated basically as little fragments. And then we get those fragments and we align them up to a reference genome. And based on how they align up to a reference genome, we can determine whether there's a mutation present or not. And then we can determine whether that is something that's significant and can actually cause an abnormal protein to be expressed. So going back to the blood, as I said, there's also the plasma component and the platelets, and that's very important for clotting. And so patients with cancer and hematological malignancies often have clotting disorders, as Professor Ireland has indicated. And so we need to sometimes look at the ability of the plasma to induce clotting. And so we have, so here we have an animation of, there's a break in the blood vessel, and we have the platelets sticking to the opening in the blood vessel and activating. And then we have other components joining in. So clotting factors that join in and then add to the ability of that, the cells there and the proteins in the plasma to form a blood clot. And so hemostasis is a fine balance. And so it's a balance between wanting your blood to clot when there's damage and not wanting your blood to clot and not having strokes, for instance. So it's very important that we measure the ability of blood to form clots. And so this is the instrument that we have in the lab. And we look at the plasma to look for clotting factors. So the plasma is tested with an automated analyzer to measure the time it takes for a clot to form in the absence of platelets. So we add plasma that's been centrifuged and the platelets have been removed, and we look at the ability of the plasma to form clots. And the same analyzer can also be used to measure the activity of individual clotting factors in the plasma. So here's our team. So Lynette, who looks after our blood bank, and Olivia, who's her second in charge, Jeff, who looks after the coagulation lab, Ewan, who looks after the morphology area, Craig, who's one of the senior scientists in the transplant lab, Esther, who looks after the flow cytometry lab, Nick, who looks after our Nick and Joanna, who look after our full blood count area, Cheryl, who looks after the molecular hematology lab, and myself, who try and keep them all under control. And thank you very much.